Hi, everybody. Welcome to our lecture today, where we are going to focus on introduction to cells. We are going to look at some of the variation of the cells, as well as looking at, sorry, I'm going to go back, looking at the differences between what we consider somatic versus germline, the difference between the vocabulary diploid versus haploid, looking at the organelles, the main structures inside the cell and their function, also looking at the membrane and the role of the membrane in the functionality of the cell, and finish up the cell structure by looking at the cytoskeleton. And at the end of the lecture, we'll focus on looking at the stem cells that are found in the human body and also relate them to how they create the cell lines that we have. So first, information about the cell. So how do we define a cell? A cell is identified as the basic unit of life. So it is basically what our body is made up of. Within our body, we have about 290 specialized cells. So if you basically look at cells that are found in every organ, every part of our body, we can categorize them to 290 cells. And then again, remember that those cells are going to form our tissues. Tissues form the organs, and then organ systems, and again, the human body. Now, these cells have the capacity to regenerate, to form, again, the four major category of tissues, which is the epithelial tissue, the connective tissue, muscle tissue, and nervous tissue. Now, every time we look at the cell have certain features, and they are going to have also a specialized characteristics that allows them to only express a certain number of genes based on the need of that cell and the location within the body. For instance, if you have a cell located inside your heart, we refer to those as the cardiac cell, and they have a very specific set of genes that they are going to express, basically make certain type of proteins. And if you compare the genes that are being expressed in your skin cells, you were looking at a different set of cells. Different set of proteins that are being produced and certain genes being expressed. Now, please do keep in mind that there are certain genes that are going to be expressed regardless of what cells we have, but then the subcategory of the genes might be different from each type of cell and their specialized characteristic based on their location. This picture, I really like it because it distinguishes or shows, the variation between the cells of our body. So on the first image, what you see is actually your nerve cells. So these kind of blue structures that you see, these are what we call neurons or nerve cells, which is what most of your brain and spinal cord is made up of. The second image is a depiction of a bone, more specifically compact bone, also demonstrated right here. So these circular structures that you see are called osteons, and basically the bone cells are responsible for maintaining these structures. This one is your blood, which is kind of easy to identify. All of these little circles would be your red blood cells, which is kind of depicted right here. And then this last one is a category of muscles, more specifically a smooth muscle, which is depicted right here. So again, looking at these cells, you kind of look at them and say, well, they look very different from one another. So does that mean that their internal structures are also going to be different? And the reality is no, they are going to have certain organelles, they're all going to have nucleus because those structures are essential for the survival of the cell. Now, as I mentioned previously, there are certain number of genes that are going to be expressed per type of cell. So blood cells is going to be different compared to a smooth muscle, compared to bone, compared to your nerve cells. But there are also category of cells that are going to be shared among all of them. So genes that are going to be expressed among all of them. Now, the next thing we're going to talk about is how do we categorize cells? So there's two major categories of cells, what we identify as somatic and what we identify as our germ cells. The way I talk about somatic cells is basically any cell in our body with the exclusion of our germline cells, which is basically our sex cells, either egg or sperm, depending on the gender of the individual. So somatic cells, also known as your body cells, are basically heart cells, skin cells, muscle cells, brain cells. Any of those categories are identified as somatic cells. Now, this is another important factor about them. They are identified as diploid cells, which we also identify as 2N. So if you take a look at this picture, let's say this is chromosome 1, chromosome 2, and chromosome 3. You do notice that they carry 2 copies. So this is copy 1, this is copy 2. Again, copy 1, copy 2, copy 1, copy 2. So one copy comes from your mom, and the other copy comes from your dad. So because these cells always going to carry 2 sets of genetic information, both from both parents, we identify them as, again, diploid, di implying there are 2 of them. And these are, again, every cell in your body. Now, germ cells are different. Germ cells are going to be the one that are basically your typical sperm or egg. Now, we do not want the full set of genetic information in the sperm and egg. Rather, we want to only have half of the genetic information here and the other half here. So that by the time the egg gets fertilized and goes to become a zygote, which is what we see here, this is a zygote. Now the zygote would be identified as a diploid. To give you a little bit more perspective in terms of numbers, again, kind of looking back at the genes and humans and the number of chromosomes we have. In humans, we have 23 chromosomes from your mom, 23 from your dad, which gives you a total of 46 diploid, right? This picture, you notice there's only 1 copy. Again, it's either from the sperm or from the egg. So there only 1 copy. So it means you have 23 chromosomes in that individual. And again, this is identified as N or 1N. Right here is the same concept. So this sperm provides 23 chromosomes, egg provides 23 chromosomes, and at the end you end up with 46 chromosome zygote, which is a diploid. Now, when we're looking at our cells, the other thing that I want you guys to understand is that our cells are actually coming from a line of cells which we call stem cells. And I think stem cells have been thrown out in kind of the general news a lot of times. But to understand this a little bit better, stem cells are basically diploid cells. Again, kind of highlighting the diploid means 2N. And they are going to divide and give rise to various type of cells. That process of basically dividing and creating new type of cells is referred to as differentiation, or the cells undergoing differentiation. They also have the option to renew themselves through a process of self-renewal. So let's take a look at the image I have on the side. So here is the original stem cell. And again, these are 2N. These cells can undergo basically self-renewal, which is what we have here. So this is self-renewal. Or they can undergo the process of differentiation, right? And right here, they have create your lung cells. They have create cardiac cells. Don't know what this is distinguishing. And then this is your bone cells. So again, we can see variation in the type of cells that are being created through the original stem cell line, through the process of cell differentiation. Now, we have understood that we have cells that are diploid and haploid. We understood that they are coming from a cell line called a stem cell, undergoing differentiation to form their final, basically, shape or differentiate to a specific type of cells. But now let's dive into, okay, so I have a cell. What is this cell actually made up of? So one of the first thing I want you guys to understand is if you're looking at a typical cell, you have to have a plasma membrane. Now, plasma membrane is really important because it's literally setting up the boundary of where your cell is located. So in this picture, again, kind of drawing this line, that line would be your cell membrane. Now, we also refer to the cell membrane as phospholipid bilayer, which simply means it's a two-layer, right? Because bilayer, that is made up of phosphate and lipid, which just simply means fat. Now, this membrane is very important for us. Again, not only the fact that it's setting up the boundary for the cell, but it's also regulating what gets in and out of the cell. So whatever, a small or large molecule that needs to get, or particles even, or ions, or water that needs to get in and out of the cell, it must be regulated by this plasma membrane. So if a cell has a damaged plasma membrane, we just typically say that that cell is dead because it has lost its boundary and the capacity to control and regulate what gets in and out of the cell. Now, what else do we have inside our cell? I think one of the structures that's really important in terms of genetic is your nucleus. Now, nucleus, again, stores your genetic information, which is your DNA. You have this whole space around the cell or within the cell that contains a fluid called cytosol. So kind of imagine all of this space, and I'm sorry, I'm kind of drawing lines, but all of this space has a fluid, and that fluid's called cytosol. And then all of these little structures that you see, these these structures that you see around here, these green structures, all of these are going to be what we call organelles. So organelles are basically going to be little organs that are responsible for basically doing everything within your cell. So if I take the fluid portion of the cell and combine it with all of the organelles that are in that space, now I get cytoplasm. Now, cytoplasm, again, does not include the nucleus. So as I mentioned, nucleus up here, we do not talk about nucleus as part of the cytoplasm. So let's start by talking about nucleus. Why is nucleus so important for us? So nucleus contain our DNA. This is something that has been brought up and discussed multiple times so far. It stores our genetic information. And again, why do I care about the genetic information? Because it tells me what proteins to synthesize. Now, the DNA is protected inside the nucleus, and the membrane that is found around the nucleus is called the nucleus envelope. So if this is the space, so this is our nucleus as a whole, this is our nucleus as a whole. You notice the light blue structure, which is this structure right here, this darker blue line would be your nuclear envelope. And then if you kind of zoom into this picture, which is what you see here, you will have these openings that allow things to basically get in or out of the nucleus. And these little holes are called the nuclear pores. So we have the DNA protected by the nuclear envelope, but allowing things to get out, in and out, we create these pores within the nuclear envelope called nuclear pores. What else do we have inside our nucleus? We have a structure called nucleolus. So if you go to this image, you have a structure inside of here, which is nucleolus. Well, why do we care about nucleolus? Nucleolus is the structure that helps us produce subunits of an organelle. This is an organelle called ribosome. And as I will discuss in just a minute, Ribosomes are super important for us because they are the ones that are going to synthesize our proteins. So this is very important for us. We want to make sure our ribosomes are formed beautifully. Lastly, within the nucleus, you have RNA, which, as we talked about, DNA form copies called RNA, and RNAs are what we use for protein synthesis. So again, looking at this process, nucleus contains significant amount of structures that are basically making sure the cell is running properly. What else do we have in the cell? So if you take a look at this picture, here is your nucleus, and the line, the red line, is identifying your nuclear envelope. But if you notice, the nuclear envelope continues in these structures right here, which is drawn in red up to this point, and then in orangish color for this portion. So there's two segmentations. Now, they kind of look like a labyrinth to me, but however you're gonna remember it. This structure, regardless of the color, so red or orange, they are identified as endoplasmic reticulum, shorthand ER. But why do we have two structures? Part of the endoplasmic reticulum contain these little dots that you see. These little dots are basically representation of your ribosomes. So these ribosomes are sitting on the surface of your endoplasmic reticulum. And because of the presence of ribosomes, they're giving it a rough surface. Therefore, we refer to that portion, which is in pink or red, as rough ER. So, because of the presence of ribosome, they're involved in protein synthesis. The orange piece that's identified as a smooth ER receives its name from the fact that it doesn't have ribosomes, and therefore the surface is very smooth, hence the word smooth ER. Well, why is there a distinction? A smooth ER actually has a completely separate job, which is lipid synthesis. Lipid synthesis is extremely important because it's involved in a lot of the activities within the cell, including the formation of the plasma membrane. Now, when the proteins are synthesized, they need to move on to basically complete their synthesis and their folding. If anything needs to be added or removed, they need to move on and basically get processed. So when these proteins are done and synthesized, they actually exit your ER through these sac-like organelles referred to as vesicles. Now, it's not depicted here, but I will show you that in just a moment. Now, we have made proteins, we have made lipids. Now they need to undergo their final processing. And that processing happened in an organelle called Golgi apparatus. Golgi apparatus is literally a stack of interconnected membrane enclosed sacs, which is this structure. So this is your Golgi apparatus. So again, it's a bunch of membranes. That's the easiest way to say it. Now, what do they do? Why is Golgi apparatus important for us? It's a processing center for us to add sugars, and it can be different type of sugars, to our protein to make what we call glycoprotein. Glyco implies sugar or carbohydrates, or add sugars to our lipids that are coming from our smooth ER, forming glycolipids. So I want to again highlight this. Glycoproteins are coming from my rough ER, or more specifically, proteins are coming from the rough ER. And glycolipids, the lipid portion of it, is coming from my smooth ER. Now, at the end, the products being produced by the Golgi apparatus, or I should say processed by the Golgi apparatus, are going to be released into another set of vesicle and then basically bought off from the plasma membrane. Understand that the proteins that we have synthesized, or the glycolipids that we have synthesized, they do need to go to different part of the cell, or sometimes in the plasma membrane, or even outside the cell, depending on the need of the body. So some cell types have just a few Golgi apparatus, but if they're producing a lot of proteins and a lot of lipids, they can have many of these little Golgi apparatus and secrete, sorry, I shouldn't say have multiple Golgi apparatus. They have Golgi apparatuses, but they secrete hundreds upon hundreds of these little vesicles into the environment. Now, one of these vesicles that are being produced by the Golgi apparatus is a specific organelle called lysosome. Lysosome is very important for your cell, like every other organelle we have talked about so far. So lysosome is this little vesicle or this little sac that contains, this is really cool, guys, 43 type of digestive enzymes. So automatically think about this. If you have digestive enzymes, the goal of those enzymes will be to break down or digest something. So what does lysosome do? Lysosome has the ability to dismantle bacterial remnants. So if you have an infection and you have a bacteria that has come inside your body, these cells have the ability to bring the fragments of the bacteria and literally digest or break it down. They also remove and break down the worn out organelles. Like everything else, organelles also get old, but that doesn't necessarily mean the cell is dying. So you can have an old organelle that gets combined with the lysosome and gets destroyed. Or if you have even excess cholesterol, you have it combined with the lysosome and break it down to simple fats. The next thing that we want to kind of understand in terms of the role of the lysosome is involved in what we call autophagy. Autophagy basically means you destroying your own cells. So you phagocytize yourself. And we'll talk about this later on as well. Now, imagine you have a cell, and this cell has engulfed or brought something inside itself. So in this case, the little vesicle that has come in is called endosomes. So they're coming in, they're bringing material into the cell. And when they come in, they merge with your lysosome. And they basically undergo changes and breakdown. What are the examples of these endosomes? The most common one is your LDL. This is something that you actually do a blood test when we do lipid panel. LDL, which is typically in common sense, is used as bad cholesterol. And again, this is not really a bad cholesterol. It stands for low density lipoproteins, and they are carriers of cholesterol. So it is important to again note that lysosomes can be involved in terms of killing an old cell or organelle, or destroying bacterial remnants that have been ingested by the cell, or just simply bringing cholesterol that is extra and breaking it apart and transporting it. Here is a scenario that I described to you guys. Here is the original cell plasma membrane. So this is the cell level. And then this is a vesicle coming in. Okay, so you form the vesicle. So there is unwanted material inside the vesicle. And on this side, you have your lysosome coming in. This is your lysosome. And what you do is you merge these two together and it gets digested. So here is the digestion process basically happening. For this one is debris and this one is extracellular debris. So I'm sorry, how should I clarify that? This is what's inside the cell that needs to be destroyed, let's say an old organelle. And this one is you bringing something unwanted from outside. Extracellular means from outside. But regardless of if it's coming from the outside into the cell or from within the cell going into the cell, so it's either inside the cell or from the outside of the cell, it doesn't matter. They will merge with the lysosomes and the components get broken down or digested, as you can see right here and right here. What else do we have in terms of lysosome? What is important in terms of lysosome? They are different type of cells in our body has different number of lysosomes. For instance, if we're talking about macrophages, which is a really, really important type of white blood cells or WBCs, they require significant number of lysosomes because they use it to destroy bacteria. Liver cells also require a significant number of lysosomes because they're tasked by breaking down cholesterol, toxins, and drugs. And by the way, toxins, for most cases, not always, but alcohol would be a very important toxin that your body needs to get rid of. Now, if you have any malfunction of an enzyme that can causes damage to the lysosome or even absence of certain enzymes inside the lysosome can cause medical condition. For instance, Tay-Sachs disease is a medical condition that has been inherited. So it is a genetic disease and it is associated with your nervous system being buried inside the lipid. And this is because, again, your lysosome is unable to break down the lipid and remove it. So those lipids get packed inside the nervous system. It overwhelms the nervous system, and unfortunately, in almost across the board, causing death on people under the age of 3. So it is a very very hard medical condition that doesn't really give a long prognosis to the patient. Now, the other organelle that we have is peroxisome. So a lot of people confused and said, when I look at the cell, we already have a lysosome with the job of digestion. Why do I need peroxisomes? Peroxisomes also contains several types of enzymes, which means they're probably going to be involved in a process of carrying a major function for the cell. But what do? They actually break down lipids and rare biochemicals. They also help you with synthesizing something called bile acids, which is very important for digestion. They involve in detoxification of components that are very dangerous to our survival. This is something associated with our own metabolism. So it's not something necessarily coming from outside. And they create something really healthy. So there is a huge number of these in liver and kidneys because these two organelles are responsible for detoxification inside your body. So liver and kidney are going to have high level of peroxisomes inside their cells. So there is a condition called adrenoleukodystrophy, which is again a genetic disease where the peroxisome enzymes and one of the peroxisome enzymes is absent. Now, I do want to add a quick note to the peroxisome, but which is not here. Peroxisome has an enzyme called catalase. Catalase is an extremely important enzyme in terms of our survival. We produce something called H2O2, which just stands for hydrogen peroxide. This is a metabolism byproduct, and it's very harmful. It contains free radicals that can cause damage to our cells. So this enzyme inside the peroxisome called catalase. So catalase, what it does, it takes this harmful material and breaks it down. Here's the cool thing. To H2O, which is water, and O2, which is oxygen gas. So it takes something that's super harmful to us and makes something really healthy out of it. Mitochondria, my favorite organelle. This is a typical shape of a mitochondria. It's a double membrane organelle, and it is the power plant of your cell. It is responsible for taking your food, and in most cases, it would be in a form of glucose. And through a series of processes, create ATP as your energy source. So typically from one glucose, you produce about 36 ATP. Now your body also has the ability to take lipids and make ATP. And it also has the ability to take proteins and make ATP. But which one is the most ideal way for you to make ATP? Would be using glucose. Now, again, as I mentioned, mitochondria has two membranes. There is the inner membrane that I drawn here. This is the folded one. And then the importance of that is that it holds enzyme that allows the chemical reactions to happen within the mitochondria and producing energy from that. Again, remember ATP. ATP is the form of energy, and it stands for adenosine triphosphate, which means it has kind of a backbone. And I don't want to know the backbone. And it has 1, 2, 3 phosphate. So that's your ATP. And anytime you break one of these bonds, you release energy, or it's energy that can be used basically by your cell. So again, here is a depiction of what mitochondria looks like. Like everything else that we have discussed, mitochondria numbers can also vary from one cell to another. Here is a chart that highlights the variation of the organelles that are found inside the body and their main role as well. This is a kind of a really good chart for you guys to review to make sure you understand the generic concepts. Now we're going to kind of switch gears from some of the organelles and kind of looking at the domains of life. So we as scientists understand that there are 3 basic type of cells that basically define 3 major domains. And these are your archaea and bacteria, and these are identified as unicellular prokaryotes, which means they have one cell, and they're basically cells that are very basic, if you want to understand that. Prokaryotes, they don't have, prokaryotes do not have organelles. I should say do not have membrane bound organelles. Eukarya, which includes us, has both unicellular, which means one cell, or multicellular, like any complex organism we have around us. And they're identified as eukarya because eukarya implies that they have membrane bound organelles. And again, that implies things like ER, ribosome, actually not ribosome, sorry, ER, mitochondria, I can't spell, Golgi apparatus, lysosome. So these are all identified as membrane bound organelles. So cells of domains contain globular assemblies of RNA and protein called ribosomes. So again, kind of going back to what we already talked about, ribosomes are really important for us because they are the one that synthesize proteins and allow us to express our genes. Now, eukarya may have believed to have been risen from our ancient fusion of the archaea and bacteria together. So the 2 more simplest kingdom basically combined together to form the more complex domain, which is the eukarya. So now that we have a basic understanding of the cells have created the three type of domains that we have, we're going to focus on the eukarya because that's what humans are basically come from. Now, we look at what we call organizational level. And this is what we already talked about in the previous lectures. But to just kind of highlight it again, we have the chemical level that gives rise to cells. Number of cells give rise to tissue. Tissue give rise to organs, your systems, organism, and then population. So this is basically the path of the organization if you're looking at the most simple to the most complex. Now, we have spent time talking about cells, and now we understand that cells basically combine together to make our tissues. So as I mentioned, there are four major types of tissues. Epithelium is one of the more basic types, is important in terms of protection, secretion, which basically means releasing something, absorption, which means bringing something into the body, and excretion, like removal of waste. Muscle, the most important function is contraction, which probably creating movement. Think about your muscles, your heart, intestines, etc. Nervous system is to create electrochemical signals, which basically means electricity with help of chemical signaling. And their importance is for coordination of basically, I should say control and coordination of anything that happens in our body. Now, I didn't talk about connective tissue because I wanted to kind of show you the other three main groups. And the way I think about it is if it doesn't belong to the other three groups, then it falls under the connective tissue. So for instance, if you look at it, things that are helping us support, protect, surrounds the material, surrounds like different tissues in our body, binds things together, fill in the space, they're all going to be your connective tissue. So I think about them as the leftover tissue, which is not true. They do a lot of jobs in our body. They include your bone, cartilage, blood, fat. Didn't mention all of them, but there are things called areolar tissue, dense connective tissue, dense irregular connective tissue, which is found in your tendons and your ligaments, etc. But again, understand that there are four major groups and what is the main functions associated with each of these four tissue types. Now, we again kind of going back to the eukaryotic versus prokaryotic. So eukaryotic, we said that they have the membrane-bound organelles versus prokaryotes typically lack that. Now, regardless of what we have and what type of cells we have, we will require to use these categories of macromolecules. Now, again, we talked about macromolecules briefly before, but I want to highlight it again. Macromolecules are basically large molecules that have many subunits. And you have four major groups: carbs or carbohydrates, lipids, which is basically fats and oil, proteins, and then lastly, nucleic acid. Now, this is a genetic course, so we already talked about nucleic acid at some levels, which is the DNA and RNA. The way I tell my students to process this information is think about the first two categories, which is the carbs and lipids. They are going to be your source of energy. So basically what your body is going to use to produce ATP. Now, that being said, it doesn't mean that proteins will not be used if your body really needs energy. But for most part, you're going to rely on carbohydrates and lipids for production of energy. So what is the difference between those two? Carbohydrates produce what we call energy for short period of time. Short period of time. Lipids on the other side, they're going to store energy. So these are your long-term storage. And again, this is the storage of energy. Kind of think about how your body stores energy if you consume too much food. It stores it as fat. The other thing, carbohydrates also contribute to your cell structure, as we already learned about glycolipids and glycoproteins when we talked about Golgi. Lipids, on the other hand, they form hormones. Typically, I tell my students about sex hormones like estrogen and testosterone. They form your membrane. Again, membrane was called phospholipid. So that lipid portion implies that it's made up of fat. They provide insulation. It's under your skin. Under the skin, there is a thick layer of fat that makes sure your body temperature stays inside and your core body temperature is maintained. And as I mentioned, a storage of energy. Now, again, this is a brief overview of what they do in the body. It doesn't include everything, but it gives you an idea of the importance of consuming fat in our daily basis. Proteins is the next category. If I want to simply say, what do proteins do in our body? I would say that proteins do all of the functions of the body. Now, what are those functions look like? They help us contract. They help us with our movement. Think about any type of movement you create, yet it be your smooth muscle, intestine, the iris of your eye, the heart pumping, you move your arm or your leg, walking, dancing, any type of movement comes from proteins. They're also important in terms of helping you form blood clots. There are proteins inside your blood called fibrinogen that helps you basically clot your blood. If you have low fibrinogen, that may lead to significant bleeding. It also forms a bulk of your connective tissue. One of the most important proteins that we have is collagen. It's found in your hair, nail, skin, and they basically have a very important protective role. But one thing that I didn't mention on the protein, and it's actually its own category, it's supposed to be a little bit forward, is enzymes. Every enzyme, every chemical reaction that happens inside your body happens because of enzymes. And all of the enzymes are proteins. Every enzyme that you have has a protein component in it. And these are basically, again, allow you to carry out chemical reaction at a much faster rate. Hence the word catalyze. Catalyze means it's speeding up the chemical reactions. So enzymes are extremely important for your survival. Now, as I mentioned before in the earlier lectures, how do I know what proteins to make and how much of it to make? And for that, we rely on our last group, which is nucleic acid. We rely on our DNA. We look at the sequence of our DNA and we understand how much of a protein and how, when and where do we make that protein. And all that information is coming from our DNA. Now, again, remember, we use DNA to make copies of it to form a main category of DNA, which is called mRNA. And we'll talk about the other types of RNA in later portion of the lecture. This is a picture of a animal cell that we have. Plant cells are slightly different and they do have some additional organelles as well as the cell wall. But because we're talking about human genetics in this class, we will look at an animal, typical animal cell. So you can identify some of the main structure. Starting from here, you have the nucleolus, you have the nucleus, you have the rough ER, we have the a smooth ER. We have our Golgi, peroxisome, and lysosome. And then we also have our mitochondria. And that's basically the main structures. And then these little dots would be our ribosomes. Now, if you notice, we also have these kind of lines and fragments that are different thickness. These are what we call your cytoskeletons, which basically, as the name implies, are going to be the skeleton within your cell. Cyto means cell. Cytoskeleton means basically the skeletal structures of a cell. Now, before we talk about the cytoskeleton, I did want to take a moment to show you this process that's associated with secretion, or basically release of the material outside your cell as your cell produces material. This is called making milk. This process, I guess I should say, explains the process of making milk. Again, it's a secretion coming in. It's a regular process that does happen, where the organelles basically, again, produce something and then releases to the external environment. So in the first thing we have, again, the genes are going to encode for milk proteins. And they're producing, I should say the DNA of this protein is first undergoing transcription. So transcription produces the mRNA, which is basically the copy. The mRNA is going to leave your nucleus, and you can see it from this picture. The mRNA, which is the green strand, is leaving your nucleus and are getting introduced to your rough endoplasmic reticulum. Now, rough endoplasmic reticulum is important for us because they allow us to synthesize our proteins. Now, proteins is made. I need to also make some fat, because milk also includes fat, and it does that on the smooth ER. So now we have some proteins and we have some lipids. These structures are now going to move to your Golgi apparatus, and then Golgi apparatus basically process that and sends it out through these vesicles. Right, so they basically process the fat, they process the proteins, and then when they're done, they pack them into these vesicles, and that's brought off from the Golgi apparatus. And you can see that by looking at these pictures. So basically, this is what they're talking about. Now, after that, these basically, again, vesicles are going to move to the plasma membrane. They merge with the plasma membrane, and then they get released into the outside. So here is basically a molecules right here. These molecules have come in. So imagine this is your protein section. And on this side, we're depicting the lipids that were produced by the smooth ER. And it is coming down. You form your plasma membrane. You form your fat droplets. You bring it to the membrane. You wrap a membrane around it, and then you release it to the external environment. The important thing that I want you guys to understand is that the lipid droplets does have that membrane around it in the form of vesicle, because most of your body is water, and those lipid droplets will not like to interact with it. So it is important to have that plasma membrane around it to make sure that it basically is protected in a sense. And I put the product protected in kind of a quote unquote. It's not a protection, it's just they don't like to interact with water. And again, those are get released to the external environment of the cell. Now, as we talked about just right now, I said you put this plasma membrane around your fat droplets, but that plasma membrane is also around the cell. So if you're kind of looking at, sorry, I'm gonna kind of go back. If you're looking at this drawing, this layer right here, this layer, and it can go all the way around, is what we call our phospho, and I'm going to keep writing it so you guys can recall the information. Phospholipid bilayer, which means, again, two layers. I'm going to say two layers that's made up of phosphate and lipid. So what is that exactly? So this is a membrane very specific to our body. It does contain these two components, as I say, phosphate and the lipids in a form of fatty acid chains. I do want you guys to understand that the phosphate heads, we shall kind of draw them as these little circles. And again, there's two lines, two layers. These are phosphate, and they love to interact with water. They have no problem with water. So we call them hydrophilic, which literally translates to water loving. The fatty acid tail, which is the lipid portion, there are two of them on each of these phosphates. And then draw them like this. So this is the fatty acid. And fatty acids do not like to interact with water. So they're called hydrophobic. Again, hydro implies water. Phobic, think is about phobia, fear. So these are either we refer to them as water hating or water fearing. They're afraid of water. So they're kind of hiding in the middle when there's little water to no water involved. Phosphate have water here and they have water here. And they don't care because they love water. Now, in addition to having these phospholipid bilayers, which is the fatty acid tails and the phosphate heads, we also have proteins that are embedded in those membranes with specific characteristics. And we also have carbohydrates that are embedded inside that plasma membrane. Here is the depiction right here. So again, the circles depict your phosphate heads. This is the fatty acid tails. The purple structures depict your proteins, and the little green hexagonal structures are the carbohydrates. Proteins and proteins within the lipids, what is their job? What do they do for us? Why do we need to have them in the membrane? They help us identify the cell as a very specific type of cell. These are usually referred to as antigens or AG. They are helping with the transport of molecules in and out of the cells. So they're acting more like channels for movement of ions like sodium, potassium, chloride, calcium, etc. And they also keep out the toxin and pathogens to make sure, basically, again, any unwanted material doesn't get in. Let's move on to more detail about the plasma membrane. What do the plasma membrane do for our cells? So we already kind of talked about some of the essential functions, making sure keeping the boundary, controlling what gets in and out of the cells. But what else does it do? They also help us with the cell-to-cell communication. So again, if you have a cell and you're looking at that cell within the tissue, you will have thousands upon thousands of cells that are sitting right next to each other. And you want to be able to have some sort of communication between these cells. And these communications happen because of the connections between these cells, where they basically have mechanisms and more specifically receptors that allows these cells to communicate with each other through a sequence or cascade of chemical reactions. Now, this process, this passing of the signal from one place to another, is referred to as signal transduction. And again, they move from the importance of this signaling is that if one cell sends a signal, it connects to the receptor, and that receptor is going to bring that message into the cell from the outside environment. Another way that this plasma membrane and the components within that is important is because it allows the cells to stay attached to each other. And that adhesion, that connection between the cells is also very important. Now, as I talked about I wanted to see what my next slide was. We have talked about the cells. We also want to finish up talking about the cytoskeleton, which is, again, I mentioned this before. Cytoskeletons are basically, again, cyte means cell. So these are a skeleton of the cell. This is what holds the cell, basically, in terms of shape, in terms of where your organelles are located. If something needs to move from one location to another, it is extremely important to have the cytoskeleton. Now, we categorize these cytoskeletons, which are truly made up of proteins based on their size. We will have these proteins that combine to form relatively very thin structures called microfilaments, and they're made up of actin. You have kind of a little bit, I guess, thicker cytoskeleton that are called intermediate filaments, which is kind of easy to remember, and they're made up of more than one type of protein. And then the third one that actually looks like a tube or a cylinder, empty cylinder, is made up of a protein called tubulin, and we refer to those as microtubules. To help you visualize the three types, again, microfilament is relatively small. Intermediate filament gets a little bit bigger, and then here's your microtubules that kind of looks like, again, kind of like a cylinder with proteins forming the surrounding of the cylinder or the wall of the cylinder. Here's looking at the size, again, 7 nanometers or microns versus 10 versus 23. So significant increase in terms of numbering. So why do I care about microtubules? Microtubules are important for me because they form structures like cilia, which are these tiny hair-like extensions. So these are hair-like extensions on the cell surface. In this case, the cells are epithelial cells. And sorry, I didn't include the other things. The microtubules and intermediate filaments are important in helping keeping the 3D shape of the cells and positioning of the organelles, as well as moving the organelles from one position to another. So I'm going to add that, moving organelles. Okay. Now, we have talked about the cells. We have talked about the structure, the nucleus, the cytoskeleton, how we categorize cells to either prokaryotes or eukaryotes based on what you found inside the cell. And we also talked about the plasma membrane of the cell, which controls what gets inside and outside the cell. It basically sets a boundary for the cell and also allows for cell-to-cell communication and adhesion. We're going to finish this lecture by talking about our stem cells, which was also previously mentioned. So stem cells, again, it's important for you guys to remember these are 2N or diploid cells. We have two paths that these cells can take. One is that they can renew themselves and basically maintain the cell lines. That's through the process of self-renewal, or they can undergo differentiation to create new cells for different tissues. Now, we create what the differentiation process creates cells called progenitor cells. The progenitor cells do not have the capacity to self-renew. So you notice that they only go into one direction, versus a stem cell has the capacity to renew or differentiate. And again, as you can see, these progenitor cells on these pictures have specialized to become white blood cells. They could have as easily become heart cells or muscle cells or brain cells, depending on what type of genes is activated and expressed in those cells. In this case, again, they differentiated to become white blood cells. Now, cells differentiate down cell lineages, going from the stem cells to progenitor cells. And then as they go to progenitor cells, they differentiate further and further to form very specific categories of those 200 plus cell types that we have inside our body. The stem cells as well as the progenitor cells are described in terms of what they have in terms of potential, basically what they can become. If they're considered totipotent, they can rise to form any type of cells in our body. They're called totipotent. Pluripotent, they have fewer possibilities, so they can just go to become any type of cells. And then multipotent have only a few developmental choices. So you're basically becoming more narrow and narrow. This is I guess, the most popular because you can become anything. Then you lose some of those options as you go through differentiation. So you become pluripotent, and then you even further distinguish this and you become multipotent. Many, if not all, of our organs, like your heart, brains, spinal cord, liver, etc., in an adult body has both of our stem as well as the progenitor cells. Here again, kind of showing you the pathway. We have a fertilized egg, which is basically the original stem cell. So at this stem cell, you can kind of follow it down. You can self-renew, which is the arrow going back, or you can start to differentiate. Here is where you form the first line of progenitor cells. And then again, as you're going down further and further, you lose the ability to differentiate to any type of cells. So you're going from totipotent to pluripotent to multipotent. Now, where does the stem cells actually come from? Where are these stem cells resources or sources within our body? They can actually come from embryonic stem cells, which is what we call the tissue that's been created in a lab dish using what identify as inner cell mass, or ICM, of an embryo. An embryo is basically a fertilized zygote or fertilized egg. A second category of stem cells are called induced pluripotent stem cells. As soon as you hear the pluripotent, which means they have more limited options in terms of differentiation. So these are somatic cells that can be reprogrammed to differentiate into several types of cells. They cannot become every type of cells. And then the third type is adult stem cells, which is tissue-specific or somatic stem cells. So you basically find them in a specific tissue, and that's all they become. Now, researchers do study stem cells to understand about our developmental stages. They can understand about what treatments we can actually use for different diseases based on the source of the disease or medical condition. Again, here is a chart that shows you those three types of stem cells and where they are coming from. Here is a kind of a depiction of, again, what we have talked about in terms of cells. I like this image because it shows that you provide, on the top one, you're providing donor stem cells, and then donor stem cells or your own cells can be reprogrammed to form a specific type of cells. And this type of cell, basically, this is a pluripotent. And in this case, they differentiate to either blood cells, muscle cells, or nerve cells. And then you can put those back into the person's body. This is really cool. It is controversial in sense, but it's also a very big step in terms of helping us using donor stem cells that are from somebody else to create various type of cells that can be donated to a patient with medical conditions. So what are some of the applications? Something I just mentioned. It's important in allowing us to discover and develop new drugs. It allows us how the disease develops. So looking at it at the earliest stage, especially again, we're looking at genetic disorders. It allows us to create tissues and organs from basically a stem cell line and then use them for transplant purposes. Again, many tissues are produced in the lab and then transplanted into the person's body. And then stem cells can also be introduced for the purpose of reprogramming proteins. So that's about it for our lecture today, guys. If you do have, again, any questions about today's lecture, please don't hesitate to contact me. I'll be happy to help you guys with any concerns you have about the lecture. And please make sure you complete the coursework, including the Canvas post. I will see you in your next lecture.